So in section 4, we're going to talk a little bit more about the structure of cardiac muscle tissue and compare it to skeletal muscle tissue, which you talked about in A&P1. So cardiac muscle cells, they're going to be striated. They're short, branched, and interconnected. Typically have one nucleus. Sometimes they'll have two. They have huge amounts of really large mitochondria. And if you remember the function of this organelle from A&P1, it's the site of cellular respiration whose end goal is to produce huge amounts of ATP. So this is going to help provide high resistance to fatigue and allow the heart to beat continuously without tiring. The cells are organized with sarcomeres, again, just like we saw with skeletal muscle tissue, meaning that they have the Z discs, the dark bands or A bands, or the light bands called I bands. While the T tubules are still present, they are a lot wider and we don't have as many of them. And the sarcoplasmic reticulum is also there, but it's a lot simpler and we don't form the triads that we saw with skeletal muscle. One of the defining features of cardiac muscle tissue, especially histology-wise, is the presence of intercalated discs. So the intercalated disc is the point that is linking two of the cardiac muscle cells together. And in the intercalated disc, there's going to be desmosomes, and these are going to help hold the cells together during contraction. And there's also gap junctions, which are just little holes between the cells that allow the ions to flow between them. And this is going to enable the heart to function as one single coordinated unit. So this is just a visual kind of talking about what we just mentioned. So here's our intercalated disc. It's this dark line right here. We have intercalated discs where we have cardiac muscle cells joined together. So this is showing you a segment of one cardiac muscle, and connected to it is another cardiac muscle cell. So the point of the connection is an intercalated disc. If we were to zoom in on an intercalated disc, we would be able to see this gap junction right here. That's our pore that allows ions to flow for communication. And we can see our desmosomes right here. They're going to help keep the cells together during contraction and relaxation so that we don't tear them apart. This is a good visual to kind of show you that we still have a lot of the same structures that we saw with skeletal muscle tissue. So we still have our T tubules, this little gray thing extending off of the plasma membrane, and we still have our sarcoplasmic reticulum, which is this blue web-like structure whose function is to store calcium ions. So all of that's the same. It's just not as elaborate as we saw with skeletal muscle tissue. So some more similarities between cardiac muscle and skeletal muscle would be that contraction is still triggered by a depolarizing action potential. And this action potential is going to travel down the T tubules, and as it does, it's going to cause our sarcoplasmic reticulum to release the calcium ions. Remember, the sarcoplasmic reticulum is really just functioning as a calcium storage bank. And once the sarcoplasmic reticulum releases these calcium ions, it's going to initiate excitation-contraction coupling, and the calcium is going to go bind to troponin, leading to the sliding filament model that you previously talked about. So all of that's the same that you saw with skeletal muscle tissue. However, there are some key differences. So some cardiac muscle cells are self-excitable, unlike our skeletal muscle. Our skeletal muscle required neural stimulation. We had to say, hey, we want to contract this muscle, and we did it. Cardiac muscle cells, we cannot initiate their contraction. We can influence the rate of their contraction, but we can't signal from our nervous system to actually cause contraction. So there's actually two main types of cardiac muscle cells, or myocytes. We have our contractile cells that are responsible for producing contractions, and we also have our pacemaker cells, which are going to spontaneously depolarize and set our heart's basic rhythm. Again, we can influence the rhythm, but we can't initiate the contraction. So these pacemaker cells can activate the entire heart without any nervous system input, because again, they're just spontaneously depolarizing. So another important difference is that the heart contracts as a unit, which we just talked about, meaning all the cells are working together to pump blood efficiently. Whereas skeletal muscles, we can contract them independently. We're not going to contract them necessarily all at once. Additionally, the cardiac muscle contraction is going to rely on both intracellular and extracellular calcium. When we talked about skeletal muscle cells, our only source of calcium was from our calcium storage bank, which was the sarcoplasmic reticulum. So we still have the sarcoplasmic reticulum releasing calcium, but we're also going to pull calcium from the extracellular space. The depolarization is going to open slow calcium channels, allowing calcium to enter in from outside of the cell, which is then going to trigger the sarcoplasmic reticulum to release even more calcium. So this is a process that's not used by skeletal muscle. Another difference is that the heart depends almost entirely on aerobic respiration for energy. We already talked about how cardiac muscle cells have more mitochondria than skeletal muscle, so they cannot survive without oxygen. We talked about skeletal muscle and how they can function anaerobically for short periods by undergoing lactic acid fermentation. We can't do that in cardiac muscle. So interestingly, this makes cardiac muscle tissue extremely resilient, but also highly vulnerable to oxygen deprivation, and this is sometimes what can lead to a heart attack.